Welcome to your lesson on Discovery of DNA, Groundbreaking Experiments. All right, really, we have about 100,000 different proteins in the body, and they do all sorts of different things. Sometimes they are used for structural things, like you've heard of muscle fibers being made of proteins. You know now that you've got proteins that are used for transportation in cell membranes. You know that there are proteins that are used for enzymes. You're going to learn in our next unit that proteins are also used as hormones. Like they're all over the place. We got about 100,000 different proteins just in the human body alone. And they are incredibly complex and an incredibly diverse group of proteins. They're all very different. And you already know that if you change the shape ever so slightly, you're going to change its function. So every single one of them with their different shapes and the different levels of complexity all carry out very different biological functions. Okay, so they thought because your proteins look very similar to your parents' proteins, that in the cell, when the egg and the sperm get together and they fertilize and you become you for the very first time at that moment of conception, then you've got all the proteins from both parents. And they thought that's just how things happen. Parents give proteins. But as they started to look more into it, they started to kind of figure out what the heck was going on with DNA and with the RNA. Now, back in 1869, they had no clue what the actual structure of DNA was. They didn't even know what DNA was. This guy, Freddy Griffith, who's a Swiss biochemist, he was messing around with some pus cells, which is got to be a gross job. But they discovered that inside the nuclei, there's a ton of something that wasn't a protein. Now, that's basically all they knew. There was something in there, some kind of a substance that wasn't a protein. So Freddy called it nuclein. Had no idea what the composition was, no idea what it was, didn't know what it did, just knew it wasn't a protein. So it's something else in there. Much later on in the 1950s, when we actually figured out the chemical composition of DNA, then the nuclein's name was changed to deoxyribonucleic acid, which we talked about in our first unit, and we'll talk about more in the next lesson. Fast forward to the 1940s. 1943, you have Joachim Hammerling, who's a Danish-German marine biologist. He is actually working at the University of Berlin while he's doing this study. And if you think what's happening during 1943, Berlin must have been a very interesting place at that point in time. 2 years before the end of World War II, and very much so the height of Hitler's reign and Nazism in Germany. So Joachim, Mr. Hammerling, is hanging out with some single-cell green algae. This is what they actually look like. You can see the size compared to this thumb here. In his first experiment, he took this guy right here, and what he did is he chopped off the little cap. And he was left behind with just the stalk and this foot here. Now you can visibly see, when you're actually looking at these guys, you can see that the nucleus is down inside the foot. You can see it in each of these pictures here as well. So he chopped off the cap, and then let it be. And he found that this new cap was formed. But when he did the opposite experiment, when he chopped off the foot and just left the cap, nothing changed. You didn't get a new foot growing. So that told him that there's some kind of material, genetic material, something that has instructions to say, grow new cap, as it does in this picture here, is sitting in the nucleus. Because when the nucleus is gone, nothing happens. You can't grow a new foot. All you can do is grow a new cap. So the information must be down in this section. There's something special about the nucleus. Okay, so he did a second experiment. It's a little bit more elaborate, and he takes two different versions, because you don't just have this round cap shape, you also have a petal shape. So this is my version of it. I'm sure it looks nothing like this, but this is how great my artistic talents are. All right, so basically he did the same thing, cut off the cap, but this time when he took the foot with the nucleus in it, he took just a portion of one of these caps above here, not the cap itself, just the part of the stalk, and he attached it to the petal foot, and then he just let it be. And what resulted was this weird in-between version, or an intermediate cap shape, which was a combination between disc type and petal type. Then he cuts off this cap again, so he's just got the petal foot again, no part of the disc stalk. And what do you know? A normal petal grew back. Okay? So why is that all so important in that second thing? Well, it means that the instructions for making petal were definitely remaining in the foot the entire time. The instructions were there. It's just the message got mixed up when I had other messages from the disc available in the stalk. So I had petal information coming up here, and I had make disc information coming up, and they blended together to make this section. How is that possible? Well, we're going to learn more about DNA and RNA as we move forward, but the basic idea would be that in the nucleus, when you produce RNA, so RNA would be the copy of the DNA, it is sent out into the rest of the cell where ribosomes attach and make a protein. So there must have been leftover information proteins for growing disc type. There must have been proteins left behind or RNA left behind when he chopped off this stalk and stuck it on here. So you had the brand new information from the petal being developed, and you had the old leftover information from the disc cap being read as well, so that the combination of those made this weird in-between shape. But when you chopped off that section, you no longer had information from the disc, and you were able to grow the normal petal cap back again. This is not proof that DNA is doing this. This is just proof that the nucleus is important. Okay? Now, the problem with the nucleus is that it has DNA and RNA and proteins in there. So it still doesn't tell you whether proteins or DNA or RNA are responsible for having genetic information. But it is key that you understand that Freddy Griffith just knew there was something different that wasn't a protein. Joachim Hammerling knew there was something special about the nucleus, and neither of these men knew that DNA was the genetic material. They still both thought proteins were super important, that parents gave you proteins rather than your DNA. Okay, so what is this term transforming principle? Let me dive into that. There's another Freddy. His name is Freddy Griffith. Notice we've backed up to 1928. So this is quite a bit before Hammerling is doing his acetobularia experiments. Okay, now he's right about this time, they're developing vaccines against all sorts of things. If you think back, 10 years earlier was the 1918 Spanish flu, which just decimated pretty much every country on earth that came into contact with Europe. Estimated to have killed about 50 million people. That's not 50 million people sick. That's 50 million people dead from the Spanish flu. Very, very scary because there was no immunity developed. It was a brand new flu that popped out. So they got a vaccine that they're working on, and they're trying to do this vaccine against pneumonia, and stumbles upon this idea of transformation. I'll get into what that means in a second. So here's what he was doing. He was using two different versions of the bacterial version of pneumonia. There are viruses that also cause pneumonia, but this one is the bacterial version. So Streptococcus pneumoniae. And there's two different versions. There is the S strain. S because it's actually got a smooth outer coat. You can see that underneath the microscope. And this one causes disease, so we call that pathogenic. The R strain, the one that looks rough on the outside, the R strain, though, is non-pathogenic. Not all bacteria cause disease. Bunch of bacteria do nothing to you. They have no harm for you. They don't produce any products that hurt you. Vast majority are non-pathogenic. There's only some that are pathogenic. So the deadly S strain and the no disease causing, everything's fine, R strain. Okay? So I'll come back to this in a second. Here's what he did. He took a live version, so the bacteria is alive, and he injected a mouse. What do you know? Mouse dies. Okay? He takes the non-disease causing R strain. Ding, yay, mouse lives. All right? Then he takes the S strain and he actually kills that bacteria. He heat kills it. So it's denatured, therefore it's dead version. Well, what do you know? Dead bacteria. Ding, mouse lives. Wonderful. Okay? Here's where it got weird. He takes the heat-killed pathogenic S strain, which should allow a mouse to live. He takes the live, non-pathogenic R strain, which again should allow a mouse to live. Mouse dies. What the heck just happened? Neither of those should be able to kill a mouse on their own. So there's something else going on. And he called this the transforming principle. Something was taking that R strain, which was alive, and turning it into dangerous living S strain bacteria. Much later on, we realized how that actually happens, and I'll talk to you about transformation on another day. But the basic idea would be that when you heat kill this S strain, you damage the cell, and basically what happens is the DNA from the cell becomes free floating. And if that's in the same place at the same time as the R strain, the R strain can actually pick up the DNA from this guy and it can become part of the R strain's genome. And if that's the case, then it now behaves like a smooth. So it's now producing products from that extra DNA that it picked up, is producing the protein products that cause the toxic effects in the mice. So again, we'll talk about transforming in a different day, but the transforming principle, okay, the idea of it is that something is able to change the normal R strain into an S strain. Again, this is 1928. Freddy Griffith has no idea whether it's the proteins or DNA or RNA. He doesn't know. He just knows something can do that, and that's pretty cool. 1944, so right after Hammerling is doing his experiments, but keep in mind, Avery, McCord, and McLeod are on a different team than Hammerling. Hammerling is working out of Berlin. These guys are part of the Allied forces, but they're working with Freddy Griffith's transforming principle, and they actually do, through a really neat little experiment, they actually end up identifying DNA as being the transforming principle, but the results got rejected. Scientific community was still really strongly believing that proteins were the shiznit. These were the ones that were doing all of the transforming. This is what they thought. They were wrong, but this is what the scientific community thought. So Avery, McCord, and McLeod didn't get any credit at this point in time, and other people had to keep doing experiments to try and prove it. Okay, so here's what they did. They took that same deadly S strain, and they heat killed it as well. But in addition to that, they also added enzymes. Now, they know that protein, RNA, and DNA are different. In 1944, we didn't know the structure of DNA yet. We wouldn't have proved that until 1953 with Watson and Crick. But they do know that it's there, and they are able to figure out how to damage it. So they take 3 different enzymes. The first enzyme destroys proteins. That means that the RNA and the DNA were left intact. Then the second one destroyed the RNA, so the protein and the DNA are intact. And then the third enzyme destroyed the DNA, so the protein and the RNA were intact. In these 2 cases, in both of these first 2 experiments, the mouse dies, and in the third experiment, the mouse lived. Notice what's still intact. DNA and DNA in both of these situations. If the DNA is still intact, then the S strain, when you add it with the live R strain, will kill the mouse. But when the DNA is damaged, so it doesn't matter if the protein and the RNA are good to go, if the DNA is damaged, then there's nothing to transform the R strain and the mouse is going to live. So this really is proof that the DNA, not proteins or RNA, was Freddy Griffith's transforming principle. But again, they didn't get the credit for that. Who did get credit was Hershey and Chase in a really neat little experiment. These guys are using viruses now. Now, they're not using the type of virus that could hurt you. They're using the type of virus, a T2 bacteriophage, which is basically a virus that infects bacteria. So if you had bacteriophage T2 on you, it would be fine. It couldn't enter your cells. It couldn't damage you. It only has the keys to get into a bacterium. So we call it a bacteriophage because it is a bacteria eater. Phage means to eat. When you see a bacteriophage, please don't think it's a bacteria. Remember that it's a virus that eats bacteria. It's a killer of bacteria, not a bacteria itself. So what are they made out of? Well, the simplest bacteriophages, like this T2, have a piece of DNA on the inside, and that DNA has the instructions to create more viruses. It's basically the blueprint for viruses. And then it's surrounded by a protein coat. Now, of course, at this time, Hershey and Chase hadn't proved that DNA was the actual code. That's what their experiment is going to try to prove. They want to know, since it's just DNA and protein, and they're just comparing the two, between the two of them, which one of these actually has the information to create new viruses. So they get their sample of bacteria in the first experiment, and they just allow the viruses to infect. Now, what should happen is the viruses infect, they inject the genetic material, and then you should see them grow on the inside, boom, 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 boom. Next thing you know, after they've replicated thousands of viruses, they're going to burst out of that cell or perform lysis, and then that's going to spread to the next cell and so on and so on. Okay. But before they allowed the virus to do that, they did 2 separate experiments. They know that proteins contain sulfur atoms, but they don't have any phosphorus. They also know that DNA has phosphorus, but does not have any sulfur. So that's really key. So what they did is they tagged the viral proteins with radioactive sulfur, knowing that the outside cap is the protein part. They wanted to know, was any of this protein injected into the cell? And then same thing with the DNA with radioactive phosphorus. So was any of the DNA injected into the cell? And here's what they found. They found at the end of the thing, there was radioactive phosphorus inside, but there was no radioactive sulfur inside. So that's proof. Viral DNA did get inside, then the viruses were made. In this case, viral DNA got inside, but the viral proteins did not get inside. Therefore, DNA has to be the hereditary material. Because again, new viruses were produced in both cases. So this experiment only shows that the DNA entered the host and that the protein coat did not. So there's your solid proof then that Freddy Griffith's transforming principle must be DNA. And that, my friends, is the end of this note.